Hello and welcome to week 6, where we're going to discuss the contents of chapter 3 and talk about some sampling techniques. Now, thus far we have talked about what should go into your chapter 1, your introduction, your background and significance, your limitations and assumptions that you're going to make, definitions of terms, especially technical jargon that someone may not understand, and then you have a summary and overview. That summary and overview is where you basically tell them, this is what I'm going to research, this is why I'm researching it, so now let me tell you about my research. Remember a couple of key components. This is a very formal document and it's all written in the past tense. So everything is past tense. You said the researcher surveyed the participants, the study was conducted, everything is past tense. Just a little bit to go over. Chapter 2 was the review of literature, also called the lit review sometimes. And we talked about the different places you can get information for your review of literature and how you can put that into different components or different headings in your review to support the reason that you're doing this, because there's either not enough literature out there or you have literature that says this is a valid concern and it needs more information done on it. Last week we talked about what goes on when you do your human subjects review. Just a quick recap for assignment number 3. A couple of people had a few questions about how to determine the correct answers for some of these. So question number 3 seemed to be one that stumped a few people, because the question said, what are the 4 ways to show the need for undertaking a study? Remember that significance is what is the research term for showing the need. So what are the 4 ways to show the significance for undertaking the study? Sorry. So that means you have to draw a conclusion. You have to be able to realize what need and significance are synonymous in this case. And then remember, limitations can be in 2 categories. They can be categorical or methodological. And also the assumptions. The assumptions are things that we take basically at face value. We say we're going to assume that the population that you are using is the correct population. So it's an assumption. So those are just a couple of things that people had a little bit of a problem with. But let's go ahead and jump into the lesson now. So the first thing we want to have a quick review on is about variables. Remember that an independent variable, those are the ones that we manipulate, that we move around. An independent variable is the one that's going to make that dependent, the one that depends on what's happening, change. So here's a little chart to help you remember what we do. That the independent variable is the one that we know that we're going to be making, that's the cause, and the dependent variable is going to be the effect. That was always something tricky for me to try to remember when I first started in my graduate studies, because for some reason my mind always wanted to think of them backwards. So this chart helps a little bit. Here's another way to think about it. The independent variable, say caffeine, influences and gives this effect on the dependent variable is, I guess, the direct intake of caffeine is going to influence the number of words that you can recall in some type of testing situation. So you look at this again and you see we've still got the independent variable and the number of words recalled, but there are other variables that sometimes are called confounding variables, or just variables that are out there that can affect the outcome of your dependent variable. So let's talk a little bit about chapter 3. Chapter 3 has some major components to it. And the first one that we want to talk about is your introduction. You know, remember that You're going to talk about the type of research you want to do, and your subsections are going to be described under there. You're going to talk some about the population. What is the population that you have decided that you are going to use? You're going to talk about your research variables. What is going to be your independent and what are your dependent variables. And then you're going to talk about the instrument, how you designed what you used. If you make a survey yourself and you make this instrument and you administer this survey, or if you have a questionnaire, you do an inventory, any of those kind of things, how did you collect your data? And you can also explain the instrument if it was something that someone else designed. There are multitudes of tests and surveys out there already. In fact, in the ODU library, there is a journal that lists all the tests that are available. Usually, if you use an instrument that was designed by someone else, there's a cost involved. But you know that it is has been vetted and it's a valid survey, so sometimes that's easier than doing it yourself. But those are the main things that go into your chapter 3. So just like I said at the beginning, just remember that your this chapter is your dissertation, and your whole paper is a very formal and it's a very scholarly document. So it has to get a lot more scrutiny than, say, a regular homework assignment. The biggest thing is detail. You have to try to anticipate any kind of question, no matter how strange it may seem, that your reader might have, because you're familiar with the content. You are invested in it. You have researched it. You've worked on it. To you, it's very familiar, but you have to explain the things to you that are, to your reader that to you are common sense in very minute detail, to make sure that nothing is left where someone doesn't quite understand exactly what you did to get the results that you got. So some other components of chapter 3 is that you talk about your field or your lab procedures that you used. You also talk about your methods of data collection. Then you have where you analyze your statistics, which is always the fun part because it's a lot of data. It's sometimes a lot of tables and figures. And then you summarize everything that was in chapter 3. So you notice a little trend here. Every time you do a chapter, you have a summary of it at the end. So let's talk a little bit about a population. What is a population? A population is all of a select group. And a sample is just a subset of that target population. Very important concepts here to remember. Population is your biggest group, as you see right here, so it gets the big N. Your sample is just a little chunk of it, so it just gets the lowercase n. These are easy to confuse, so the easiest way to think of it is the biggest one gets the uppercase letter, and the little subsection gets the lowercase letter. So what we want to do is that we want to ensure that we have a quality sample when we get our sample so that we can make inferences on the entire population. So if you have a poor sample, you're going to have a poor inference. So you want to make sure that you have one. You want to have what we call an unbiased sample, which means every person in that population has an equal chance of being selected or included in the sample. So that is called a simple random sample, and we're going to talk about some more sampling techniques in a moment. Now this chart right here, even if you look at the date, you'll say, wow, that's kind of old, but this really hasn't changed. This tells you if you have for your big N, your entire population, what the size of your population is and how many you need in your sample for your sample to be relevant so that you can make correct inferences. So obviously, the smaller your population is, the larger sample size you need to be able to generalize your data. And you see once you get to 100, it's 80 needed, 500, it goes down to less than half. There is actually a chart, and You can find these charts everywhere about population or sample size. This is just one reference to have a chart like that. So we talked a little bit about the random sample, and that's typically what people use when they are doing a sample. It's representative of the whole, like I said, and everyone has an equal opportunity to be selected. So selecting a random sample, there's different ways you can do it. You can get your whole population together and pull numbers from a hat. There's such a thing as a random number generator. Most of us have been, maybe we want to say, victims of a random sample when you're doing classwork face-to-face, or when you're in a group meeting and they say, everybody count off 1, 2, 3, 1, 2, 3, and then you get put in a group based on whatever number you said. So that is an example of just having a random way to divide people into different groups, or in this case, into a sample. Then we also have what's called a systematic sample, and it's a kind of a random sample, but what they do is they select every nth number. So they decide, I'm going to go by 3s, as in this example, or maybe I'm going to go by 5s, or I'm dependent upon how big your sample is. But it's another way just to pick every particular number that's in your entire sample. Then we get to the really interesting ones, and this is kind of a fun one. It's called a stratified random sample. And what you do is you take these subgroups that are in your entire big population and put them in what they call a strata. Actually, strata is plural because it's a Latin word, so it's kind of like data. The singular is stratum, so one is a stratum, more than one is a strata. And this way you get a truer picture of the whole population, and it guarantees that you're going to have more equal representation in these different strata. I mean, the most common one that everybody can think of is you have a population and you divide them into male and female. That is one way to divide them into subpopulations and then pick people from each of those. So let's look at this example right here. Suppose we have 12,000 students. 63.3 are female and 36.7 are male. 20% of both of them wear glasses. And you see this says, see page 110 in the text for more information. This is more information about stratified samples. So let's talk about how we're going to do this problem. 12,000 students, 63.3 female, 36.7 male. So we go up here, we have this population. This is how many we have. So if you take 12,000 and you multiply it, now I got to remember what the numbers were, I turned the page. You multiply the 12,000 by 63.3, you will get 7,596 female. So you can either easily subtract it from the 12,000, or you can take the 36.7 and multiply it by 12,000 to get the number of males. And then the problem said that we have 20% of both wear glasses. So 20% of the 7,596 is 1,500, and 20% of the 4,404 is 881. So now we have these stratified samples. Originally, we had 2 strata right here, but then we broke it down into 4 different strata for this sample. So we could pick a few from the glasses female, a few from the non-glasses female, some from the glasses male, and some from the non-glasses male to get a better even distribution of all that are in our sample. So let's talk a little bit about census data. Census data is another way to pick information about your group, and you conduct a census and you find out your ratios and the people that you have. So for this census data, we have 8 boys to 10 girls. That's your ratio. Everybody remembers ratios from math. So that's 8 to 10. 10% of the entire population held college degrees. We need 180 people or 180 participants for the sample. So what would be the characteristics of this sample? So if we know we need 180 and it's 8 boys to 10 girls, think about this. What would be the percentage? So we're going to have, if we have 180 in our sample, we'll have 100 girls. 10% of that 100 would be 10 with parents with college degrees. We have 8 boys to every 10 girls. So if we have 100 girls, obviously we have 80 left, so it's got to be the boys. And then you take the 80 times 10, and so you 10%, and you have 8 parents have college degrees. So this is another type of a stratified random sample. Then we talk about some other ways that sometimes we do samples, and sometimes they're okay, but it depends upon really what you're researching. There is a sample called a convenience sample, and I believe also in your topics in your textbook, they also call that an accidental sample. It's one that, people are conveniently available. It could be, you have a focus group with your local Lions Club. It's practical because the people are there. It could be that you use a group of athletes because you're on the basketball team, and so you use that as your sample because you see these people all the time. The problems with this is that usually when people are in groups like that, they're not considered to be generalizable to the larger population. So you want to avoid that if possible. But sometimes, depending upon what your research is and what you're trying to do, it can be the only practical way that you can do what you want to do to research your subject. Then we have what they call non-random samples, and these are ones where, like the title implies, that is not really random, and this is a hard thing to say, it's a purposive sample. It's a sample that you select because these people have special qualifications of some sort. I mean, you may choose people who are recent graduates of a PhD program. That's not a random sample because you are technically looking for only people who just recently completed a PhD. But you select them for that or because you have some prior evidence of representation. So one of the problems with this is that, you could have a little judgment error here. So if you're going to pick a sample like this, make sure that you find enough literature to support the fact that you are doing this. And then we start talking about sample size. Everybody would love to say, I was able to talk to 10,000 people and these are the results I got. So this has got to definitely be something that's important. But when you start thinking about your sample sizes, sometimes when you have a really small sample, it makes it easier for you to analyze the data. It makes it easier for you to determine exactly what your data is going to tell you. But if you don't have a very large sample, then it may not be as accurate because you don't have as much diversity and as much difference in people when you have a small sample. And then if you have a huge sample that's overly large, it is going to be more accurate because you have sent this information out to a much larger subset of the population. However, it is going to be very costly to do that, and it's going to be very time consuming. Even when you're talking that you have 200 or more that are in, you might have a sample, let's say you have a sample of 250, that's still a lot of data to manipulate and to work with. So you need to think carefully about how much time and how much effort and how much you really love doing statistics to generalize your population from the size sample you need. It's all going to depend upon what size, what you're trying to research and what your population is. If your population is a large number, you have a relatively large sample size that you need to use. So then we also talk about another type of sample that we can do, and it's called volunteers. And, you know, you hear a lot about people volunteering for medical studies, clinical trials, things like that. You know, there's and psychological studies and things like that. You know, these kind of patients are usually very, very cooperative, and, you know, sometimes they are compensated for volunteering. They do have, though, very specific characteristics, and it does make your data somewhat different when you get the information. You know, they're going to be more like a purposive sample, because if you're doing a clinical trial on interstitial pulmonary fibrosis and you say, I want people who have this disease, they're already a specialized subset of a larger population. So it's something that, unless you were doing something in the medical vein, it's something that we try to avoid using volunteers, because volunteers sometimes are, you know, considered a captive audience, and you know, they volunteered, so sometimes you're not really sure why they volunteered, whether they have their own agenda and will skew your data when they answer your questions. Here's some more things about volunteers, and there's a reference right here to Rosenthal and Rosnow, and that's a study that you can read to learn more about using volunteers. You know, you may get better data because volunteers did volunteer, but it's kind of a tricky thing to think about. So then we start thinking about, how are we going to collect the data that we need? First of all, any instrument you use, whether you develop a survey yourself or you use one that's already out there, you have to make sure that it has validity. So writing a survey, we think, oh shoot, that's easy. We do those all the time. You know, you're always answering surveys. You get them in your email. People say, answer these questions, or you do them at work. They say, we want to get interest in whether we want to have an employee health lounge or something, and they send out a survey. But in scholarly research, writing a survey is very complicated, because you have to make sure that your questions are valid, that they don't have any inherent bias in them, that they're not slanted toward a particular way of answering them. And then you have to ensure that they're also reliable. Are you really asking what you think you're asking, or can that question be interpreted in different ways? So there's lots of different things we can do. We can do a test, you can do a survey, or you can do interviews. So validity, like I said, it deals with whether the instrument's appropriate to what you're doing, and you have to work really hard on the questions and make sure that they are really asking what you're trying to get a true response to. Validity is also concerned with whether your survey is really going to measure what you think you're asking. You know, sometimes, and I'm sure we've all done this, you've answered questions on a survey and you say, well, all of these are kind of asking the same thing over and over again, and you wonder how valid it is. We got to make sure, and you'll learn this in other classes as you get further into your research, you'll learn ways to write questions and understand how questions should be written so the bias is out, and that the question or your instrument that you developed really measures and does what it's supposed to do. So the next thing we want to talk about is what's called construct validity. And construct validity is the overarching quality with all the other measurements of validity labels falling underneath it. It's used to determine how well a test measures what it is supposed to measure. In other words, is the test constructed in a way that it successfully tests what it claims to be testing? Then we have what they call content validity, and this is sometimes called the logical or the rational validity. And this is the estimate of how much a measure represents every single element of a construct. So, for example, SAT math section is intended to measure math skills. If all of the questions are only about subtraction, then what it would really be measuring is subtraction skills, not your mathematical ability. Then we have something called criterion-based validity. And this relates to the results of a particular kind of test you've taken. Sometimes, you take pre-employment tests as a predictor of how well you will be retained in that position, or your performance on the GRE is a predictor of your success in a graduate program. So I'm sure all of you are painfully familiar with that particular type of validity. Then we have internal validity. We have to talk about, did an experimental design make a difference? The treatments, did they make a difference in a particular instance? How much can we pull from this particular thing? And so we go back to the variables. Did the independent variable, the X, the homework, really produce a change in the dependent variable, which are your grades? This is usually only a relevant type of validity when you're trying to explain a cause and effect. The independent, the cause, making the effect on the dependent. So you have to control for, we talked about this at the very beginning, those extraneous or confounding variables that could happen. If you read this example, they talk about a company that has 8 plants. So when you check the HR records of those 8 different plants, those who get paid the most produce the most widgets. So you read those sentences, can you with a surety and say that these results are valid? Can you conclude that if you get higher pay, you're going to have greater production? No, that is a faulty inference, because there could be confounding variables out there. One plant may have higher pay for everybody. It doesn't matter how many widgets they produce. One plant might have better equipment. They might have better people that train the people who make the widgets. They might have better management to motivate the people in the plant. So external validity is asking the question, when you're thinking about it, to what population settings, treatments, or variables can I take this effect that I found, and can it be generalized? What is the relevance? What do the findings concerning the effect of this one particular thing, my X, have beyond my particular experiment that I've done? So you can strengthen the external validity of your design if you describe the population into which the results are going to apply before you do the experiment. So that goes back to our limitations and our assumptions that we talked about a couple weeks ago. So when we're talking about external validity, we have to remember those things, those extraneous or confounding variables like we talked, and we have to be real careful. So some of the things that we talked about earlier, we have to make sure when we're selecting our sample that we don't have any bias. Was there any effect of any kind of pre-testing going on? What kind of procedures did we use? And if there's more than one treatment, if you find that maybe some parts of the sample had more than one treatment, how is that going to change your overall experiment? So these are things that you have to be careful when you're doing your research. So what we want to really talk about is, does your instrument produce the same results consistently? And that's a term that we use in research called constancy. And So there are ways to check for this, and there's one thing that's kind of a interesting little twist. It's called inter-rater reliability. And of course, I find one of these examples kind of crazy, but I left it in here anyway. You know, one of the things that's inter-rater reliability could be based on baseball umpires. Think about it. What percentage of umpires call a ball or a strike? This other one, you have to be kind of old to get this inference. That's why I started to take it off. But there was a lady called Bo Derek, and she was in a movie. I can't even remember the rest of the name of it, but it had to do with 10. And that's where back in the dark ages, they came up with this rating scale of how attractive one might find a female. So this was called the Bo Derek scale. And so some form of inter-rater reliability would be what percentage of men rate a woman's attractiveness on the Bo Derek scale of 1 to 10. Then we also have what's called test-retest, and what it does is it measures test consistency, reliability to measure the test over time. What you do is you give the same test twice to the same people at different times to see if the scores are the same or very close to the same. And then we have what's called parallel forms, two forms that are slightly different in the formulation of the form, but they measure the same construct. And then we have what they call internal consistency, which is a way to measure, you could say, I am a happy person, I'm usually in a good mood, and I am not an unhappy person. Those all mean the same thing. Listen to it again. I am a happy person. I am usually in a good mood, which would imply that I'm happy. And C, I am not an unhappy person. So these all measure the same thing, and consistent responses should result in similar results. So to go back over what's in chapter 3 again, remember you have the introduction, which is untitled. You're going to talk about your population, and then you're going to talk about your sample. And usually you say, my big N, which is the population, was, maybe 500 people, and my sample consisted of 217. I believe that's the correct number for a 500 sample population. And you explain your population, who you chose, what characteristics are in that population, and how you chose your sample. Was it a random simple sample, or was it a stratified sample? Then you talk about the variables that you're going to run into during your research. And then you want to talk about your instrument. Is it one you developed yourself? Is it one that you have used that someone else has developed? You're going to talk about how you determined that it's valid and it's reliable. Then you're going to talk about how you're going to collect your data. The next step is to talk about the statistical analysis, how you're going to take the data you have collected and analyze it. And then the last step is to write a summary of all the things that are in this chapter, what your population is, what your variables are, what kind of data, you're collecting it by a survey, and what you hope to prove when you actually analyze your data. So this is it for this week. If you have any questions or comments, please don't hesitate to contact me. You can call me, or you can send me an email, and I try to read them as quickly as I can during the day. Sometimes they slip between the cracks. So if you don't hear from me within 12 hours after sending one, send it again, because sometimes I have so many emails coming in, sometimes they slide between and I don't see them, but I don't intentionally overlook any email. So I try to respond as promptly and as quickly as I can, because I know in a graduate program, when you have a question, you need that question answered quicker so that you can keep moving forward. All right, and thank you for listening to this lecture.